In Chapter 8, we're going to talk about photosynthesis. Photosynthesis, I would argue, is one of the most important biological reactions on Earth. It is the source of the energy that we consume because we're heterotrophs. But it also has changed the Earth in a large number of ways. Photosynthesis is also super critical economically. We think about agriculture, that's all really based on photosynthesis. And as our world changes, which it inevitably will, climates have always changed. We had ice ages, now we don't have ice ages. We're going to need to adapt our agriculture to it. So a better understanding of photosynthesis, where it works well, what different types of photosynthesis plants do, that'll teach us more about how we can adapt our agriculture and perpetuate our society ultimately. So we'll talk about photosynthesis in a broad view, and then we will talk about a series of reactions called the light-dependent reactions, and then the second part, the light-independent reactions, that's where we're actually building our sugar. We're going to focus primarily on plant photosynthesis. There are slight variations on this in microbes. There's anoxygenic photosynthesis, things like that. We'll touch briefly on those, but this is mainly focused on plants. So let's look at photosynthesis as a whole. You can see a common theme here, right? In all of the pictures I've been showing, we have the color green. We'll talk about how that plays a role in photosynthesis as we go along. So we're going to talk about the significance of photosynthesis, not only just to the photosynthetic organisms, but to other living organisms. We'll talk about the main structures involved in there, and then talk about the substrates that go in and the products that come out of photosynthesis. So at a high level, photosynthesis is just energy conversion. It is anabolic in nature. The key piece is that it's capturing energy from sunlight. So we're taking light energy and using that to fix carbon. So I have this term here, fix carbon. It sounds kind of strange, but what fixation means is taking inorganic molecules like CO2 and sticking them together to build a larger molecule, in this case, a sugar. So carbon fixation is taking carbon dioxide from the atmosphere, which is abundant, and putting it together to build sugar molecules. Now, that's an anabolic process, right? So we're going to need energy to put those things together, and that's going to come from the sunlight. In the process, waste products are going to be made. In most of our plants and many of our microbes, that waste product is this substance called oxygen. You might not think of oxygen as a waste product, but it is the waste from photosynthesis. Now, we have very cleverly figured out a way to use that, but I don't want you to think that the purpose of photosynthesis is to produce oxygen. That is a byproduct. The purpose, if there is any, of photosynthesis is to build sugar molecules by fixing carbon. That's the main goal here. There's a whole bunch of stored energy on our planet that is the result of photosynthesis. Coal, oil, all these things. That was energy captured by photosynthesis and put into organic molecules 300 million years ago. Over time, heat, pressure have converted them into different hydrocarbons, but really are fossil fuels. They're not fossilized at all. They're just converted carbon into hydrocarbons. So we've taken plant material and compressed it and turned it into these products here. Photosynthesis is the key reaction that enables the global carbon cycle. So we have our photoautotrophs, right? They're using the light energy to fix carbon into sugar. That's really important because we're taking that inorganic carbon from CO2 and putting it into an organic molecule. Heterotrophs consume that energy in those organic molecules. So we're a bit of a parasite on the plants, right? They fix that carbon and then we eat them or eat something that's eaten them. So we can't survive without the photoautotrophs. And then we produce water and carbon dioxide in the process. That carbon dioxide gets recycled into this. So there is a cycle of carbon moving around. There are some let's call them more exotic organisms that don't really participate in the light part of this. There are chemoautotrophs, like at the bottom of the ocean and things like this, around hydrothermal vents, that can make their own energy from inorganic chemical compounds like minerals. Some of them participate in the carbon cycle. They're still carbon-based life, but they might not necessarily be fixing into sugars. The majority of these are microbes, though. Again, specialized microbiology classes is where you could learn more about these. But there are other kind of cycles going on, but the main one is the global carbon cycle here, this fixation of carbon dioxide into sugar. So photosynthesis is an anabolic reaction, and I would love for you to know this overall reaction, what goes in and what comes out of it. I don't need you to know the chemical formulas. I don't need it to be balanced. I just want you to know the pieces. So in photosynthesis, we start with carbon dioxide and water. Those both go into photosynthesis. We capture energy from sunlight, and we use that to stick the carbons together to build sugar. And in the process, oxygen is generated as waste products. That's the overall reaction, right? That's as simple as we can make it. How that occurs is somewhat complicated, but very cool. Plant leaves are super specialized structures for photosynthesis. Most plants make leaves. They have evolved specially to capture sunlight. So the plant leaf is optimized for photosynthesis. We have light capture at the top and gas exchange at the bottom, right? We said we needed light energy to come in and CO2 to come in and oxygen to go out. So they're specialized for that. Some parts of the leaf structure, they are different. So you might think of a leaf as this broad green surface, but it has many different layers. Usually leaves have some sort of waxy cuticle. One big problem for plants is water loss, right? Water is hard to come by. They can't lose it frivolously. We know that water is needed for photosynthesis, so we don't want to lose it. At the top, under that, we have an epidermis layer. I'll talk more about that in a moment because I have a personal connection to that. The palisade mesophyll, so meso means middle. So the mesophyll is where a lot of the photosynthesis happens. We have two parts of that. The palisade is specialized for light capture, right? We're capturing most of our light up here, lots of chloroplasts in these cells. And then below that is the spongy mesophyll. It has gaps and space between them. That's there for gas exchange, right? Gases come in, they float up, and they're able to make their way up to the palisade mesophyll. We then have a bottom epidermis, and we have a large number of these cells called guard cells, which guard stomata. Stomata are pores in the leaf that regulate gas exchange and help prevent water loss. So guard cells can open and close to regulate whether water is coming in or out. Now they also can control the gas exchange. So they're key in regulating this photosynthetic reaction. So here's our cartoon. Here's an actual stained sample here. You can see the palisade mesophyll up here and the spongy mesophyll down here. Here's a stomatal pore here with the guard cells around it. Now, why do I say I have a connection to the epidermis? Well, when I was a wee little undergrad here, longer ago than I would like to remember, I did undergraduate research at the University of Washington studying plant stomatal development. So we had this mutant that turned the entire epidermis of plants into stomata. Now, as you can imagine, that's not necessarily a good thing. Lots of water loss occurs. So here you can see the normal, what we call the wild type, Arabidopsis. Arabidopsis is a weedy plant, but we study it because it's really good for genetics and things like that. It grows quick. Here we have the Scream D mutant, and it's called Scream because it looks like the Scream painting with all the mouths open. It's super short and stunted, and it's like wet look, and it's got all these problems with it. So what I was doing was trying to find what we call a suppressor mutation, a second mutation that causes this not to happen. So I did find two of those. I screened thousands of plants, probably tens of thousands, hundreds of thousands, maybe even, just looking for one that turned this back into something that looked more like this. And I found two of them. And I did that probably back in around 2010. And many, many years later, in the 2020s, Finally, this is how long scientific research takes, someone picked up that work and continued it, and we got a paper out of that. I'm very proud of that. Here's me at my undergrad presentation, my crappy little poster there, but we got a paper out of that much later, so it was really rewarding to see that stuff pay off there. And undergrad research is great at getting you involved in the process of biology, right? I was a pretty poor student until I started doing undergraduate research, and then I really realized why I was learning all this somewhat boring stuff in my classes, right? It really applied in biology. So in the leaf cells, there are structures called chloroplasts. As we've talked about, these are organelles. Their structure is really important to the process of photosynthesis. The chloroplast itself has a double membrane, and then inside of it is more membrane stacked into things called thylakoids. The thylakoids stack into a granum. That's a stack of thylakoids, and it's surrounded by this stromal fluid. The key here is that in the thylakoid membranes, we have a pigment called chlorophyll. Now, pigments have color. We see a green color. I'll talk about why that happens. Pigments in biology are molecules that absorb light. That's really critical for photosynthesis, right? Because we need to absorb light energy. Chlorophyll is that pigment. The thylakoid membrane has an electron transport chain in it. We'll see. It looks very much like mitochondria. So these are endosymbionts, and just like mitochondria, they've evolved from bacteria, but in this case, cyanobacteria, the blue-green bacteria that do photosynthesis. So this will look very similar in nature and have some similar reactions, but it's doing kind of the opposite process of mitochondria. We can break photosynthesis up into two parts. I'm going to give you the now accepted proper names, and then I will tell you the less proper name in a moment for it. Photosynthesis can be divided up into light-dependent reactions and light-independent reactions. I'll talk about what happens about those in a sec, but in the past, this used to be named the light reactions and the dark reactions. That's not quite accurate because the light-independent reactions can also happen when there's still sunlight around. So it doesn't have to happen at night, but it can. It doesn't need light. We'll see why in a moment. So let's look at them in detail. The light-dependent reactions are where we're capturing the energy from sunlight, and we're actually generating some ATP and this molecule called NADPH, a high-energy electron carrier. Looks slightly familiar. In this process, we're using H2O, water, and we're making oxygen as waste. So we're splitting water up and oxygen as a waste product there. Chlorophyll in the thylakoids here is taking in the light energy and converting it into chemical energy stored in the form of ATP and NADPH. Okay, so you'll notice in the light-dependent reactions, there's nothing about sugar here, nothing about CO2. That's the light-independent reactions. In the light-independent reactions, also, of course, having a second name, the Calvin cycle, the light-independent reactions use that ATP and NADPH to fix carbon dioxide into longer molecules. Ultimately, glucose is going to be made from this. So we take CO2 and we stick it together and we produce a sugar from that. You'll notice the light energy was already converted, so this is why it's light independent here. Let's review before we finish here. So why are plant stomata mainly on the bottom of leaves? Go and pause the video and think about that. Okay, this is kind of a two-parter. We have to think about what's happening in the stomata. So they're letting gas in, but there's no reason that more gas would be better on the top or the bottom, right? Gas is gas, it's going to come in. So let's think about the other part of this, light. Well, light is great. Plants want light, but what does light bring with it? It brings us heat energy, and heat can cause water loss. So if we put the stomata on the bottom of the leaf, the light hits the top, but the bottom is actually cooler and less water evaporates or transpires through those pores on the bottom of the leaves. So bottom of the leaves, we have stomata there to minimize water loss here. All right, so we defined carbon fixation. That's kind of the key to all of this. We're taking inorganic CO2, bonding the carbons together to form larger organic molecules, sugars. Photosynthesis is doing this utilizing light energy to fix that carbon. This is an anabolic reaction. CO2 and water go in, and they make sugars, and oxygen is produced as a waste product. The plant leaf structure has evolved to be very efficient at photosynthesis, minimizing water loss but allowing gas exchange. Chloroplasts in those plant cells have the thylakoid membranes in there. That is where we have the photosynthetic reactions occurring. We'll look at them in more detail in the coming sections. Pigments are light-absorbing molecules. The pigment in photosynthesis primarily is chlorophyll. And photosynthesis has two parts. We have the light-dependent reactions that generates ATP and NADPH, produces oxygen as a waste product there, consuming water. Then we send that to the light-independent, which we use the ATP and NADPH to fix the carbon together, producing our sugars. All right, that is it for 8.1.